morning, students. In our earlier classes, we have discussed very many cycles pathway, and we have also learned that how a respiratory process, that catabolism is going on in any living system. Today we will be learning another new pathway which is going on inside any living cell, and it is playing a very important role as far as the living system is concerned, and that is called the urea cycle. And today I will start with this urea cycle, followed by gluconeogenesis, and it will be ended with glyoxalate pathway. So let us start with urea cycle. Now, urea cycle is otherwise known as ornithine cycle. The biochemical reaction which is going on in every living cells, including that animal being, and that produces urea from ammonia. This cycle is the first metabolic cycle discovered by Hans Krebs and Kurt Henseleit in 1932. In this particular cycle, what we will be learning? We will be seeing that how urea is being produced in the living system. Now, ammonia, which is released mostly by this amino acid, is breakdown in the liver. Some of this ammonia is used in the biosynthesis of nitrogenous compound, and the remaining excess ammonia, mainly present in the form of ammonium ion, is found to be toxic, and at high concentration in the living system, if it starts getting accumulated, it has been seen that it causes brain damage. So, this particular byproduct which is produced inside the cell is to be removed. And how it is removed? It is removed through this urea cycle. So, ammonia has to be effectively removed from the cell because of this toxic effect. This removal of this ammonia takes place in the following way. In case of aquatic animals, so in the beginning only I have told you that in any living system this type of urea cycle is present. So, in case of the aquatic animal, mainly these bony fishes, it directly excrete ammonia into the surrounding water, and this group of animals are called ammonotelic animals. That means these are the group of animal, they are secreting ammonia directly to the surroundings, and it is going out of the body system. In case of terrestrial vertebrates, including human, so in case of our system, what we do, how we excrete, it is getting excreted in the form of urea, and this group of animals are called urotelic animals. In case of birds and reptiles, where they convert this ammonia to uric acid before excretion, and that group of animals are called uricotelic animals. So, these are the different variations. So, what we have learned? We have learned that this ammonia can directly be released from the body, it may be converted to urea, or it may be converted to uric acid to the surrounding environment, and it is going out of the system. And when such type of metabolism, when such type of processes are going on inside the system, we are detoxifying our body from this toxic accumulation. Today we will be learning that how urea is being excreted from the system, living system. Now, if we see the structure of urea, it is the diamine of carbon, very simple structure, that is 2 amino group is attached to this C11O. In contrast to ammonia, which is basic in nature, urea is neutral and hence it is non-toxic in nature. It is easily transported in blood and excreted in urine. Urea is produced in a cycle, sequence of the reactions called as urea cycle. Its location, if we see the location of this urea cycle, then in the liver cell and subcellular location, that is, if we further classify, then we can find that it is partly taking place in the mitochondria and partly it is taking place in the cytoplasmic fluid of the cell. So, you just see, in case of glycolytic process, we have seen that glycolysis is taking place in the cytoplasmic fluid. Mitochondria is taking place in the this TCA cycle is taking place in the mitochondria, but you see this urea cycle which is taking place in partly in the mitochondria and partly in the cytoplasmic fluid. So, this is the particular cycle. Now, here if we see in comparison to the other metabolic pathways, what we have already learned that glycolytic pathway, when I discussed, I told that this process is the 10 steps process. In case of TCA cycle, I told that TCA cycle is the 8 steps process, and in each step we have seen that the role of precursors are immense. We cannot ignore. In this particular urea cycle also, we have seen that precursors are playing a significant role in the 5-step process of urea cycle. Now, these precursors are carbon dioxide in the form of bicarbonate, this nitrogen. How nitrogen nitro groups are coming? One is directly derived from the ammonium, that is NH4+, and second one is coming from the amino acid, that is the aspartate. ATP molecules are playing a significant role in this urea cycle. In case of that respiratory process, we have seen that how ATP molecules are generated when this catabolic process is going on in the living system. Here we will be seeing that how ATP driven process is also simultaneously going on in the living system, and urea cycle is one of such example. Now, here ATP is needed to carry forward this reaction. Water molecule is also playing a significant role in the urea cycle. Now, as I have told you that urea cycle is a 5-step process, and here different enzymes are playing a very important role in this particular cycle. If we see the different enzymes, we can find that carbamyl phosphate synthetase 1, ornithine transcarbamylase, arginosuccinate synthase, arginosuccinate lyase and arginase enzymes are playing a vital role in this urea cycle. Now, at a glance, if I give you the overview of this urea cycle, then we can see that initially this ammonium ion and bicarbonate in presence of two molecules of ATP is produced carbamyl phosphate, and this carbamyl phosphate is the first product of urea cycle. This particular reaction is taking place in the presence of the enzyme called carbamyl phosphate synthase 1. This particular reaction, when it is going on, it produces two molecules of ADP and inorganic phosphate. This particular step is going on inside the mitochondria of the cell. When we are coming to the second step, carbamyl phosphate and ornithine, which is already present in the mitochondria, it is producing citrulline and one inorganic phosphate in the presence of ornithine transcarbamylase, and here this step is also taking place in the mitochondria of the cell. As soon as citrulline is produced, citrulline is going out of this mitochondria and it is coming to the cytoplasmic fluid of the cell, and citrulline in presence of aspartate and one molecule of ATP is needed to go forward this reaction, and it is producing arginosuccinate and one AMP and inorganic phosphate. In the presence of this enzyme arginosuccinate synthase, and this reaction, as I have mentioned, that it is taking place in the cytoplasmic fluid of the cell. Arginosuccinate in the presence of arginosuccinate lyase. This is the reaction when lytic process, breakdown process is going on, and it is once again forming this arginine and fumarate, and it is taking place in the cytoplasmic fluid of the cell. Arginine in presence of water once again getting converted to ornithine and urea. Urea is going out of the system, and ornithine from the cytoplasmic fluid is once again entering to the mitochondria to carry out this cycle, and this way continuously this cycle is going on inside any living system, and urea is generated and going out of this living system. Now, I have told you the overview of this reaction. Actually, how these reactions are going on, this is a very simple pathway, and here, as I have told you the overview, and then we will come systematically each and every step and how this product and byproducts are produced, we will also see and learn. See here, this carbon dioxide and ammonia in presence of two molecules of ATP producing this ADP and PI, and carbamyl phosphate is produced. See this carbon dioxide which is donating this bicarbonate, and this ammonia, see here ammonia is there. See this ammonia in the form of ammonium ion, two molecules of ATP and one water molecule is yielding carbamyl phosphate along with the production of 2 ADP, PI and 3 hydrogen molecule. So, this is the first step of urea cycle. Now, here you see carbamyl phosphate synthase enzyme is playing a significant role in this conversion process. Now, when this is getting converted, it is this carbamyl phosphate is produced. This carbamyl phosphate in presence of ornithine is converted to citrulline, and the enzyme which is playing a significant role is the ornithine carbamyl transferase. So, here you see this is producing this citrulline. This process, up to this process, it is going on in the mitochondria of the cell. Here, as soon as citrulline is produced, citrulline in presence of aspartate in and the enzyme arginosuccinate synthase is getting synthesized in presence of one molecule of ATP and producing arginosuccinate. Arginosuccinate, when it is produced in the presence of the enzyme arginosuccinate lyase, is once again splitted into arginine and fumarate. This fumarate is, we know that this fumarate is one of the byproduct of TCA cycle. So, this fumarate is entering to the TCA cycle, and this arginine, which is another product of this particular cycle, goes forward, and in the presence of this enzyme arginase, it releases this urea from it and another product ornithine. This ornithine is once again entering to this mitochondria of the cell. Now, let us come stepwise to this particular reaction, that how these reactions are going on and how the intermediate products and byproducts are produced. In the first step, we have learned that bicarbonate and ammonium ion combine to form carbamyl phosphate. So, we have already learned this carbamyl phosphate formation, where this bicarbonate, that is HCO3 minus, which is contributed by this carbon dioxide, and from this ammonia, this ammonium ion is coming. So, these two products, when they are coming, this bicarbonate, you see this is the bicarbonate, and when ATP molecule, I told you that 2 molecules of ATP are needed, this is 2 molecules of ATP. So, first molecule ATP is coming and it is donating its phosphate group and it is producing carboxyphosphate. You see this phosphate group is donated by this ATP and ADP production is there. That means, this bicarbonate is getting converted to carboxyphosphate. Now, here when carboxyphosphate formation is there, then ammonia in the form of ammonium ion is coming and playing a significant role. Now, this ammonia is coming and it is forming the carbamic acid. That means, this carboxyphosphate now once again become this removal of phosphate group is taking place and it becomes carbamic acid. When this carbamic acid is formed, another molecule of ATP is coming and donating its phosphate to this carbamic acid, and ultimately you see this carbamyl phosphate is formed. You see this is the phosphate and this is the carbamyl group, and carbamyl phosphate is produced, which is the end product of this first step of urea cycle. Now, if we sum up, then this step comprises the 3 sub-steps where phosphorylation of bicarbonate forms the carboxyphosphate. This is the first step. Reaction with ammonium ion, with the carboxyphosphate to form carbamic acid. This is the second step. And finally, once again this carbamic acid is getting phosphorylated, and finally phosphorylation of carbamic acid to carbamyl phosphate is taking place. So, we can see that this reaction, what we have seen, that though it is showing a single step reaction, but in reality it is a complex reaction where 3 sub steps are needed, where bicarbonate is converted to carboxyphosphate, carboxyphosphate is converted to carbamic acid, and once again ultimately carbamyl phosphate is produced. Now, when this carbamyl phosphate is production is over, then this second step, this is the second step. In this step 2, what is happening? This carbamyl phosphate is reacting with the ornithine. See, this is the ornithine which is there in mitochondria, is coming in contact with this carbamyl phosphate, and it is producing the citrulline. Now, here in this second step, carbamyl residue is transferred to ornithine forming citrulline. That means, this is the carbamyl residue. These carbamyl residues are getting transferred to this ornithine group, and this ultimate result is the formation of citrulline. This reaction is catalyzed by ornithine transcarbamylase enzyme. Citrulline is passed into cytoplasm via the transporter, the transport system which is there. This citrulline, now also this citrulline which is produced is in the mitochondria of the cell, and this citrulline is now coming through transporter to the cytoplasmic fluid of the cell. So, now this transportation of this citrulline is taking place. So, in this step, what we have seen? We have seen that carbamyl phosphate, this carbamyl group is being transferred to ornithine and citrulline is formed. Now, in the next step, what is there? That aspartate is condenses with this citrulline forming arginosuccinate. Now, if we see this citrulline, which is the end product of the second step, in the step 3, what is happening? This citrulline in presence of aspartate, that condensation reaction is going on in presence of one molecule of ATP which is producing one AMP and inorganic phosphate, and arginosuccinate is produced. This reaction is catalyzed by arginosuccinate synthetase enzyme. Now, here we can see that here this transfer of this group is taking place, and this amino group of this aspartate is going and getting binded, and the new group is getting connected with the citrulline producing arginosuccinate. This is the condensation reaction. Arginosuccinate in the next step is getting cleaved. Now, this arginosuccinate is once again producing this, separating this arginine from the fumarate. Now, you see here this, it is that step 4. In the next step, what is happening? This arginosuccinate in the presence of lyase enzyme. arginosuccinate lies, it breaks this bonding, and you see here this arginine, it is one amino acid arginine. See here this amine groups are getting separated, and fumarate, which is one of the byproduct of TCA cycle, is being released, and this fumarate can enter to the TCA cycle for subsequent utilization. And in the last step, what is happening in step 5? What is happening? This arginine, in the presence of the enzyme arginase, is getting converted to urea and ornithine. Now, hydrolysis of arginine generates urea and ornithine. Now, see here this group is being separated and urea is produced and ornithine is once again going and entering to the mitochondria for urea cycle. This reaction is catalyzed by this arginase enzyme. Ornithine is transported back to mitochondria for the next cell cycle. Now, if we see this urea cycle, it can be linked with the citric acid cycle. As I have already mentioned you that urea cycle, during this urea cycle, we have seen the generation of fumarate, which is one of the intermediate of TCA cycle. Each cycle though operates independently, but are connected depending on the transport of intermediates. Fumarate gets converted to malate in cytosol by fumarate dehydrogenase, that is one of the isozyme, and then it is transported to mitochondria where it takes part in that TCA cycle. So, in the mitochondria, this fumarate takes part for citric acid cycle. Aspartate in mitochondria can be transported to cytosol and it takes part in the urea cycle. Now, here whatever I have told, if I sum up, then we can find that you see here carbamoyl phosphate in the presence of ornithine is producing citrulline. As soon as citrulline, see this is the cell and this is the mitochondria and this is the cytoplasmic fluid. So, citrulline is produced, citrulline is getting transported out of this mitochondria to the cytoplasmic fluid. This citrulline is now producing arginosuccinate. Arginosuccinate, when it is cleaved, it is producing arginine and fumarate. So, this fumarate is entering to this TCA cycle back to this mitochondria. Fumarate may get converted to malate and this malate can once again come and enter to this TCA cycle, and this particular reaction we can tell that aspartate arginosuccinate shunt for citric acid cycle, which is a bypass reaction, and here this arginine which is produced is releasing urea and ornithine is transported into mitochondria and this urea cycle is getting completed, and this is all about this urea. Now, coming to this regulation, the enzyme carbamoyl phosphate synthetase 1 is allosterically activated by N-acetyl glutamate. This is one of the very important product which is controlling the activity of carbamoyl phosphate synthetase 1. Steady state level of N-acetyl glutamate is determined by the following, that is the concentration of glutamate and acetyl CoA, that is one way, and another is the arginine which is considered to be the activator of N-acetyl glutamate synthase enzyme. So, these are the two important regulator. If we see this acetyl-CoA and glutamate, we can find that in the cell acetyl-CoA is produced with fatty acid oxidation and beta oxidation. So, here we can get acetyl-CoA, and glutamate is one of the amino acid which is present in the cell, is undergoing reaction with the enzyme, with the help of the enzyme in acetyl-glutamate synthase, and it is producing the enzyme called N-acetyl-glutamate. This N-acetyl-glutamate, and here arginine is also playing a important role to catalyze this reaction. Now, this N-acetyl-glutamate is playing a significant role as it is the regulatory enzyme for carbamoyl phosphate synthetase 1. Now, this product undergoes several unknown reaction, and ultimately it is coming and it is just activating this enzyme, and this bicarbonate and ammonia in the presence of this enzyme is getting converted to carbamoyl phosphate, which is the initial product to start the urea cycle. And this way this N-acetyl-glutamate is controlling this or accelerating the rate of carbamoyl phosphate production in the cell. Now, if we see the energetics, then we can find that ammonia, carbon dioxide, aspartate, 3 molecules of ATP, 2 molecules of water gives rise to urea, fumarate, 2 molecules of inorganic phosphate, AMP, and pyrophosphate. And thus, in the entire cycle, we can find that ATP, 3 molecules of ATP are used up. It is needed to carry forward this reaction. 2 molecules of ATP is being used in the first step, and 1 molecule of ATP is used during the formation of arginosuccinate, and that is here it is the energy driven process. Now, if we see the urea cycle disorder, then we can find that an urea cycle disorder or urea cycle defect is the genetic disorder caused by a deficiency of one of the enzymes in the urea cycle, which is responsible for removing the ammonia from the bloodstream. That means, here with the defect, that accumulation of toxic substances in the form of ammonia is there inside the living system. The nitrogen accumulates in the form of ammonia, a highly toxic substance, and is not removed from the body. Urea cycle disorders are included in the category of inborn error of metabolism. Now, how to know? It can be, this disorder can be recognized by very many ways. We can measure some of the end product in the plant, and as we have already learned that this urea cycle is taking place either in the mitochondria or in the partly in the mitochondria and partly in the cytoplasm. So, here what is there? If this N-acetyl-glutamate synthase enzyme is not there, then it is called N-acetyl-glutamate synthase deficiency. If this deficiency is there, then ammonia concentration in the blood is going up. Carbamoyl phosphate synthase 1, and if this particular enzyme is not there, then also it is called carbamoyl phosphate synthase 1 deficiency, and here also this ammonia concentration is going up. ornithine transcarbamylase, this is another enzyme, and if the disorder is there, then we are calling ornithine transcarbamylase deficiency, and here it is resulting in the higher concentration of ornithine, urea, orotic acid in the blood. In the cytoplasmic, when we are considering the cytoplasmic enzymes, then arginine synthetase enzyme, and if it is not there, its deficiency is called citrullinemia, and here citrulline concentration in the blood is getting up. Arginosuccinate lyase, if not there, if it is deficient, then it is called AL deficiency or arginosuccinate acid urea deficiency, and here citrulline arginosuccinic acid concentration in the living system is going up. If arginase enzyme is deficient, arginase enzyme is not there, that is called arginemia, and here arginine concentration in the body is going up. Now, to treat such type of patient, for treatment what we have to do, we are doing here, the patient who are suffering from such type of diseases, we have to give a controlled diet where this protein intake should be minimum, where because there should not be much protein that can generate excessive ammonia, which is formed out of these protein molecules. This protein restriction is used in conjunction with the medication which provide alternative pathways for the removal of ammonia from the blood. Frequent blood testings are required to monitor the disorder, optimize treatment, and frequently hospitalizations are necessary to control this disorder. At the most extreme cases, a few liver transplantation has been taken place successfully to cure this disorder. Now, if we come to this particular disorder, and if we see this urea cycle, we can find that these amino acids are playing a significant role in this particular cell cycle, in this urea cycle. And if we see the different families, that is C4 family conversion into oxaloacetate, and if we see, we have seen that N-acetyl glutamate is playing a significant role in operating this carbamoyl phosphate synthetase 1. So, that enzyme which is there, that glutamate is playing a significant role. So, if we see inside the cell, how we can tell this aspartate and alpha ketoglutarate. Aspartate is the amino acid and alpha ketoglutarate is one of the intermediate product of TCA cycle. When they undergo some reaction, they produce oxaloacetate and this glutamate, which is playing a significant role in that supplying that N-acetyl glutamate formation. If we see that C5 family conversion into alpha ketoglutarate through glutamate, we can find that glutamine, proline, arginine, histidine, they all are amino acids, they are converted back to glutamate. When glutamate is there, glutamate is getting converted to alpha ketoglutarate, and this way this alpha ketoglutarate and glutamate is playing a significant role to operate this urea cycle. Now, coming to this another important amino acid which is also playing a significant role in this particular glutarate production. We can find that this conversion of histidine into glutamate. Histidine is also another important amino acid which is present in any living system, and it has got one imidazolium group, and this imidazolium group is present in histidine, which makes it unique from any other amino acids, and when it undergoes some reaction, ultimately we can find that this glutamate is produced from this histidine molecule, and this way this glutamate concentration in the cell is being maintained by very many pathways, from where this urea cycle or the intermediate, this glutamate, which is one of the important intermediate for this urea cycle operation, and this way this urea cycle is being operated inside any living system. Now, coming to the another topic, that is the gluconeogenesis. Now, here in my earlier class I have already discussed the synthesis of glucose, that glucose is being produced, and I have told that how glucose to glycogen and glycogen to glucose production is taking place, or this type of conversion, that excess glucose is there, it is converted to glycogen. Body is in need of glucose, glycogen is broken down to this glucose molecules, and this way this glucose generation is there from this carbohydrate moieties, that I have already discussed in my last class. But today I will be discussing that another pathway, that how glucose can also be produced from non-carbohydrate source. Now, see, the gluconeogenesis is the synthesis of glucose from non-carbohydrate precursors, that is pyruvate, lactate, glycerol and amino acids. So, here we can see that not only this carbohydrate, but the non-carbohydrates are also playing a significant role as far as this glucose production inside the cell is concerned. It is one of the two main mechanisms the body uses to keep blood glucose level from dropping too low, that is when blood glucose is dropping down, it is called hypoglycemia. The other means of maintaining blood glucose level is through degradation of glycogen, what I have already discussed, and how glycogen is being chopped, broken down to glucose, that we have already learned. Now, today we will be learning that from non-carbohydrate source how glucose is being generated. In animals, the gluconeogenesis pathway is for the most part the reverse of glycolysis. There are substitute or bypass reactions for the irreversible steps of glycolysis. Sometimes what we have learned in glycolysis, glucose is converted to pyruvate, but we can say here that pyruvate is one of the precursors for gluconeogenesis. So, from pyruvate, when glucose is produced, it is just opposite to that of this glycolytic process, but in actuality it is not totally like that, because some of the reactions which are taking place and which are non-reversible, irreversible in nature, those type of reactions are bypassed in gluconeogenesis. Now, let us learn that what are those bypass reactions which are there in gluconeogenesis. Now, here as I have told that pyruvate glucose is the gluconeogenesis. Now, if we start from the end product of glycolysis, that is pyruvate. Now, pyruvate in presence of in the presence of the enzyme pyruvate carboxylase, and one molecule of ATP is being required, and this bicarbonate is needed to convert this thing to ADP. Now, gluconeogenesis begins in the mitochondria with the formation of oxaloacetate through carboxylation of pyruvate. This reaction also requires one molecule of ATP and is catalyzed by pyruvate carboxylase. This enzyme is stimulated by high level of acetyl-CoA, that is production in beta oxidation in the liver, and inhibited by high level of ADP. Oxaloacetate then converted to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxylase. So, pyruvate is getting converted to oxaloacetate. Oxaloacetate is then in the presence of carboxykinase enzyme. This enzyme is converting this oxaloacetate to phosphoenol pyruvate, and one molecule of GTP is converted back to GDP, and one molecule of carbon dioxide is being produced in this particular reaction. That means, this is the decarboxylation reaction which is going on while conversion of oxaloacetate to phosphoenol pyruvate. This is first step, that is the bypass, which is not there in case of glycolytic process. In the second reaction, if we see the second bypass reaction of gluconeogenesis, then we can find that fructose 1,6-bisphosphate. In the presence of this enzyme, fructose 1,6-bisphosphatase is converted back to ADP to ATP. That means, from this fast phosphate, that fast carbon of this fructose, which is phosphorylated, this dephosphorylation is going on, and from the fast carbon phosphate group is being taken up, and ADP to ATP formation is taking place, and fructose 6-phosphate is produced. This particular step is also otherwise known as rate limiting step of gluconeogenesis. In the another bypass reaction, where we can see that glucose 6-phosphate is once again dephosphorylated, and it is converted back to glucose in the presence of this enzyme glucose 6-phosphate. Here also we can find that this molecule of ADP with uptake of this phosphate group is converted back to ATP. Now, the final reaction of gluconeogenesis is the formation of glucose, which occurs in the lumen of the endoplasmic reticulum, where glucose 6-phosphate is getting hydrolyzed by this glucose 6-phosphatase enzyme, and glucose is being produced as a final resultant product. Glucose is shuttled into the cytoplasm by glucose transporters located in the membrane of the endoplasmic reticulum. And this is the bypass reactions, and remaining that the other reactions which are there inside this are same as that of this glycolytic process. So, if we see this reversal of this particular reaction, we can find that this pyruvate is getting converted to glucose, and here is this bypass reactions, and these bypass reactions are different from that of this normal conventional reaction. Now, if we see the glycolysis, gluconeogenesis and pentose phosphate pathway, the main regulation of this pathway occurred in connection with this glucose 6 phosphate, and this pathway interact with at least 2 other pathways. Regulation of the pathways around glucose 6 phosphate is important. Glucose 6 phosphate and or fructose 6 phosphate are allosteric inhibitors of hexokinase, that is depending upon, or glucokinase, that is depending upon the tissue. Insulin activates glycolysis, glycogenesis and pentose phosphate pathway, but reduces gluconeogenesis, and these are the some of these differences and similarities. If we see the stoichiometry of gluconeogenesis, then we can find that 2 pyruvate, from 2 pyruvate 1 molecule of glucose is being produced, and this reaction is thermodynamically this is possible and feasible. In contrast, the stoichiometry of this reversal of glycolysis is thermodynamically unfavorable reaction, where delta G is coming positive 84 kilojoule per mole. So, here also pyruvate is getting converted to glucose. So, these are some of these differences in gluconeogenesis and glycolytic process. So, these are this bypass reaction, this fructose 6 phosphate to this glucose formation, fructose 1,6 bisphosphate. Here this is one of this reaction, phosphoenol pyruvate is another bypass reaction, and this glucose formation is the third step, which is not similar to that of glycolytic process, and here is the dissimilarities of gluconeogenesis and glycolytic process. If we see that gluconeogenesis is taking place in the liver, and in the muscle glycolysis is taking place. Now, glucose is getting converted to pyruvate to lactate, and then this lactate is being enter to this liver cell, and from lactate to pyruvate, and pyruvate to once again glucose is produced in the process through the process of gluconeogenesis, and this glucose is once again coming to this muscle cell of the body. Oxaloacetate utilizes the cytosol of this gluconeogenesis. Cytosol for gluconeogenesis is formed in the mitochondrial matrix by carboxylation of pyruvate. Oxaloacetate leaves the mitochondria by a specific transport system in the form of malate, which is reoxidized, and oxaloacetate is formed in the cytosol. So, this is the pyruvate to oxaloacetate to malate, and once again this malate is coming to the cytoplasmic fluid, and malate to oxaloacetate is produced, and this way mitochondria and the cytosol, these reactions are going on. This is, as I have already discussed, this is the reversal of this glycolytic and gluconeogenesis process. Now, if we see the similarities and dissimilarities of this glycolysis and gluconeogenesis, we can find that here 3 enzymes which are participating in the glycolysis process is different from that of this gluconeogenesis. Hexokinase, here it is in glycolytic process, here glucose 6 phosphatase. Phosphofructokinase, fructose 1,6 bisphosphatase, and pyruvate kinase, it is pyruvate carboxylase or phosphoenol pyruvate carboxykinase. These are the enzymes. And if we see the C3 family conversion to this pyruvate, we can find that alanine and alpha ketoglutarate is producing this pyruvate. This pyruvate can also be produced from this amino acid different amino acid like alanine, serine, tryptophan, glycine, and ultimately with this cysteine, threonine and amino acetone are there, and ultimately you can see this pyruvate is produced, and this pyruvate, from this pyruvate once again this your gluconeogenesis, that glucose generation may also take place in the liver cell of the body. Now, coming to the last this another cycle, that is the glyoxalate cycle. Glyoxalate cycle is an anabolic pathway which is occurring in the plant and several microorganisms. This pathway thus allows the cell to obtain energy from the fat. It utilizes acetate from fat for biosynthesis of carbohydrate. The glyoxalate cycle, whose initial reactions are identical to that of this TCA cycle. So, here we can find that this acetate is getting converted to citrate, citrate to isocitrate, and then isocitrate, that other reactions which are there, which is forming this succinate and glyoxalate, which is entering once again to this particular cycle. This glyoxalate cycle in plant is taking place in the glyoxysome of the cell. Now, see, acetyl-CoA in the presence of this particular acetyl-CoA in the presence of oxaloacetate is converted to citrate. This citrate is getting converted to isocitrate. You see, this after this reaction, it is similar to that of this TCA cycle. As soon as isocitrate is produced, it is producing succinate, and this 6 carbon is broken to 4 and 2, this glyoxalate, and here another molecule of acetyl-CoA is needed to produce this malate, and once again oxaloacetate is formed. And in this way, when we see this particular pathway, we can find that this TCA cycle, this glycolytic process, TCA cycle, urea cycle, glyoxalate pathway, this gluconeogenesis, and all these pathways which are simultaneously going on inside the cell are playing a significant role, and how we have seen that one intermediate products are being utilized for one pathway to another through the shuttle system. And this way, in very complex reactions, n number of reactions are simultaneously going on inside the cell, inside the living system. Thank you very much.